So what's the point about the garage, do you think? How many cars are parked in there? 2. So this is a diploid house. How many possible alleles are there? Are there only 2 types of cars that can be parked in there? How many possible different cars can you park in a 2-car garage? A lot of different combinations. Chevy Volt and a Nissan Xterra or whatever. And then you talk about colors and trim lines. So what was the point from last class? How many possible alleles are there of any gene? A lot. Not necessarily just 2. It's just that every individual can only have 2 alleles of every gene. I'm happy to report that after last class, so the entry quiz was testing on a lot of things that we talked about last class. The vast majority of you did really well, with rare exception. So good job, everybody, I'm happy to say. There were a few patterns, though, to briefly address. Okay. Question number 2. Some of you didn't put down C as the genotype of a diploid organism. So minus over minus. Minus is just an allele designation. So plus minus, you would expect in a diploid. Minus minus is also diploid. A over A is also diploid. What would a genotype look like of a tetraploid organism? Like a salmon. How many slashes would there be in the genotype of a tetraploid organism? If it's got 4 alleles, there'd be 3 slashes. So any diploid genotype will have 1 slash. Separates the 2 alleles that are present in that individual. So that's really the thing to focus on. There's a slash there. Here's a triploid organism and its genotype. Everybody, with maybe very rare exception, got question 3. So nice work. Heterozygous, that's easy. You look for 2 alleles that are different. Two alleles, 1 genotype. Haplotypes seem to catch a few more of you off guard. So how does a haplotype and a genotype differ? Some of these are haplotypes and some of these are genotypes in question 4. So haplotype is half of a diploid genotype. So a single allele designation, in this case a single letter or a plus by itself, would be a haplotype. That might be the designation you give to a gene that you're talking about in gametes, for example. Haploid genotype, a haplotype. And pure breeding genotypes, again, most of you got. So what are you looking for? Somebody please summarize. What are you looking for when you're looking for a pure breeding genotype?
Something that's the same.
Something that's the same. So another word for a pure breeding genotype is a same zygous, homozygous genotype. So there was one there. The GA3 over GA4 and the X1 over X2, those are different alleles of the same gene, so those wouldn't be pure breeding. Those are heterozygous. Which is not pure breeding. Any questions or concerns at this point? It's just sort of our brief review from class last time. Now, this is more review from last time. These are concepts we talked about last class. feedback to you all on the couple of things I asked you to do for last class that were due for today. I love the artistic approach. You've got fish that are heterozygous. So draw me a diagram that represents all of the different meiotic products, the gametes, that are produced over the lifetime of an individual. And these are some of the results that I got. It's unclear to me whether these represent the lifetime of an individual. Most of these are not wrong, just not what I was expecting. Most of these do accurately represent what happens when a single diploid cell goes through meiosis. That's what we've been talking about for the last couple of classes. You have a heterozygous cell, and then as it goes meiosis, you're going to make 2 big A gametes and 2 little a gametes. The vast majority of you sent that in, some version of these sorts of diagrams. That's right. So that's a single meiosis. Okay. One gamete produces one diploid myocyte produces 4 haploid gametes. And the same thing happens every time meiosis happens in that individual. So over its lifetime, what fraction of its gametes are big A? 1/2. And what fraction are little a? The other half. Exactly 50-50. Does anybody want to debate exactly? I'd love to. I want you to challenge me always. But this is one of the rare times in biology I feel comfortable saying exactly. Because we know this is how meiosis works. If you start with a diploid heterozygote, and meiosis happens, by definition, half of the gametes, every meiosis will have one allele and half the other. So over the entire lifetime of the individual, half of its gametes will carry big A's and half little a's. Might there be exceptions to this? Or do you have any concerns about this? This is the prove me wrong moment. You'll live long in infamy if you come up with an exception to this. No? Well, think about it. Email me, tweet me if you come up with an exception, because there probably is one. Here's another response to that same question. This is more what I was envisioning. All those previous ones were accurate, but this is the graph, right? 50% of gametes over the entire lifetime of an individual are big A, 50% are little a. What I would argue is that you could even represent this as a line. So you could do like a bar plot that totals 100% of gametes. You could represent the big A's as occupying 50% of the length of the bar, and the little a's as the other 50%. Over one individual's lifetime, half of its gametes are big A, half are little a. Same is true for the output of one single meiotic event. 4 gametes, 2 are big A, 2 are little a. And then there's the P word, Punnett square, that we use to incorporate all of Mendel's inferences, plus this number 6 at the bottom, which is not one that he probably understood it. He didn't phrase it as such. The idea that eggs and sperm come together in random combinations to produce generations of offspring. And it was all this together that Punnett used to devise the Punnett square, except I try to stay away from Punnett squares because they carry some sort of negative connotation. I was mentally scarred by Punnett squares up through graduate school. Because when I first learned about them in high school and when I revisited them in college as an undergraduate, I kind of understood what was going on, but I didn't fully, and it just sort of became this thorn in my side. So I try not to say Punnett square. I will, of course, because that's how we all recognize the thing that looks like this. But it's easier than just memorizing what a Punnett square is. It's really easy to apply it without actually drawing a Punnett square. So today's goals, we're going to talk a little bit about dominance. There's a whole section of the class on dominance in a little while, but it's important to bring up a couple of dominance points today. How we know which allele dominates its effect over the other allele in a diploid. And then we're going to start talking about crosses. Hopefully that's what everybody's here to talk about. Setting up crosses, how to predict the outcomes of crosses. This is one of the more applied things that you can do with genetics, especially if you're interested in plant breeding, animal breeding, or research, is trying to design how to get most efficiently the genetic outcome you want by crossing individuals together and combining their traits. So this is how you will determine the dominance relationships between two individuals, between two alleles. So we're going to cross two parents that are pure breeding for two variations, which another word for which is allele. You could replace that with allele if you want. Two different versions of the same trait. So on the left here, we're going to do two different crosses. So what are the genotypes you would have to start with? Pure breeding genotypes in the top generation here on the left. What pure breeding genotypes would you have to fill those blanks in to get an offspring that has one GFP allele and one wild type, that is, doesn't glow green allele? It's going to be diploid genotypes. Okay, so GFP over GFP, why do you say that? It's right. It's pure breeding, homozygous, right? Homozygous genotype is pure breeding. So you take a worm that's pure breeding for being green. Pure breeding means when it has its offspring, especially in the case of C. elegans, when you can self a single individual, have it produce all of its own offspring. If you take one of these hermaphrodites and you self it, 100% of its offspring are also glowing with green fluorescent protein. So it's pure breeding. What's the other pure breeding genotype? Plus over plus. Which generation is this? That blank on the left side, we're going to label the generation numbers. This is the P generation. P or P0. Either is fine. Okay. So you've got two parental generation parents. Both are pure breeding. One is pure breeding for being green. One is pure breeding for being not green. So what do we look for in the F1 generation? How do we know which of those alleles is dominant? So we want to look at the phenotype. Is it green or is it not? So if the GFP allele, if the version of this gene that is a green fluorescent protein makes a green offspring, when you're heterozygous, one of your versions of the chromosome has the gene that makes you fluorescent, one of them doesn't. What do you expect is going to happen? If you have one copy of the GFP gene and your other chromosome doesn't have that gene, are you going to be green or not? Yeah. So it turns out in this case that the heterozygote is green. So you just look in the F1 individual and ask which phenotype does it have. That's the solution to the problem. That's how you determine dominance. You cross two pure breeding parents together and you just look at the phenotype of their offspring. If the phenotype is green, then that's the dominant allele, GFP. If this F1 individual, on the other hand, has the other parent's phenotype, if it's not green, if it's plus, then that's the dominant trait, dominant allele. Cross two parents together, look at the phenotype of their kid. Which parent does the kid look like, green or not? That tells you which allele is dominant. the green allele or the not allele. So let's practice that on the other side. What parent genotypes do we have to cross to make a heterozygote at the role gene? It's going to be pure breeding, so it's going to be homozygous plus cross to homozygous minus. I guarantee you that you make a plus minus offspring genotype. Why is that? Can somebody explain really briefly? How do we know, how am I so sure that the genotype of the F1 is going to be a heterozygote in these types of crosses? What are the haplotypes of the P0 gametes? So every time this cell undergoes meiosis, what kind of haplotype is going to be produced? 100% pluses. What about this parent? All of its gametes will have minuses. So there's no other option for the F1 generation in this cross but to be heterozygous. That's why we start with pure breeding parents. We use that to know that they're homozygous. Their pure breeding equals homozygous. If you cross two homozygotes together, you get a heterozygote by definition. So what do you do with this F1? Let's say its phenotype is, it rolls. You might remember the movie. They sort of corkscrew in a circle as they crawl instead of doing the normal snake-like movement. So let's say the phenotype is that it's a roller worm. It corkscrews around as it tries to navigate a petri dish. Which allele would be dominant if the F1 is a roller? Wild type plus or minus the mutation that makes rollers? In this case, a heterozygote, if it's rolling, means that the mutation is dominant. It only takes one copy of that mutation in a diploid individual to cause that phenotype. That's the dominant phenotype. If it doesn't roll, if it just crawls normally on the plate, which allele is dominant? Better be the other one. Then the plus, the normal, the wild type allele is dominant. Any questions about dominance? We'll practice this more as we go through the next few days, and especially more when we get to the dominance chapter. Cross two homozygous parents together, look at the phenotype of their offspring compared to the parents. That tells you which of the alleles is dominant. Yes?
What if you can't tell by the phenotype?
Ooh. So by definition, so the question was, what if you can't tell by the phenotype? The definition of dominance is that you can tell by the phenotype. So it's a sort of a circular argument. It's a good question. If you can't distinguish two parents by phenotypes, then you can't assess dominance, is the easy answer to that. Then there will definitely be, this leads to a very important point, not every allele has a dominance relationship compared to other alleles. So it is not true that every allele is either dominant or recessive to another allele. In some cases there are dominance relationships, in other cases not. Does anybody know one example of that? Right, so things like incomplete dominance. So for example, blood type. Let's talk about blood type. There's codominance and there's incomplete dominance, both good examples. Flower colors is one example, blood type would be another. So there are certain circumstances where you could cross a red individual with a white individual and you make, oh goodness, now I have to try to find pink, and I'm colorblind. Is that pink? Close enough. Okay, there's no clear dominance. The F1 doesn't look like either of the parents. So there is no dominant versus recessive. Then there's the other example, for example, blood types, where you can be the A blood type, you can be a B blood type, or you could have both alleles expressed at the same time. So the A and the B are not dominant or recessive. They're both present. One is not dominant to the other. Other questions about dominance? Oh man, let's just say monohybrid cross. Try to avoid the P word. So what's going to happen if you take a homozygote and you self it? So for selfing, this is the easiest way to write the cross. It's like you're crossing a genotype to itself. You can write it out as you're performing a cross. You're using a single individual, yes. But this is exactly the same thing. So if you have a role 1 homozygous mutant, minus over minus, and you made it with itself, this is the cross you're setting up. So please predict. This is all about transmission genetics, Mendelian genetics. Please predict, what are the next generation's genotypes and phenotypes going to be? This is a worm with a roller phenotype. It's the corkscrew worm. You self it. What are all of the genotypes of its offspring? Has to be, this is the P generation parent. Its gametes are all going to be minus crossed with minus. You've told me this before. This is meiosis. Homozygous, there's only one haplotype of gamete produced. So the next generation is going to be entirely homozygous, 100% of the individuals, not just one individual. Every single F1 will have a homozygous recessive genotype. So what phenotype does that correspond with? It's rolling. It's exactly the same genotype as the parent, so it has the same phenotype. Right? The genotype of all of the kids is exactly the same as the parent. So they better have the same phenotype. They better have the same trait. That's what we would expect. So this is a monohybrid cross. Anytime we're dealing with a cross that's just involving one gene or one locus, that's when we call that cross a monohybrid cross. When we start talking about Mendel's second law, then we'll start talking about dihybrid crosses. That's looking at 2 or more genes. 2 genes in the case of a dihybrid cross. Okay. What happens if you cross the roller parent to wild type? That's the other question up there. What's going to happen? Can you predict for me what its kids' genotypes are? What one word do we use for the genotype of the F1s when you cross minus over minus to wild type? Minus over plus, or heterozygous. Het. What phenotype is that? If you've got one normal version of the gene and one mutant version, if you remember the previous slide, we already had the answer from our discussion about dominance. If you've got one mutant version of the roller gene and one wild type version, those F1s do not corkscrew. They don't roll. Which allele then is dominant? Wild type, right? The F1 doesn't roll, it's wild type. So that tells us that the wild type allele, and that's exactly what we did on the previous slide. You look at the F1, it's wild type, therefore wild type is the dominant allele. Yeah, please.
So is wild type always going to be dominant?
Not always. So wild type is not always dominant. It's a common conception, and most of the cases that we'll talk about are, but there are some rare cases, they're probably more common than you think. There are some occasional cases we'll talk about where the mutation is dominant. In fact, we'll probably spend a whole class on that. Sir?
So are we to assume that since the plus minus is a non-roller, that the negative is not sufficient to make it roll?
That would be an accurate interpretation. So having one copy of the mutation is not sufficient to create a roller. One allele insufficient. So it's another way to phrase the exact same point. That's kind of a definition of dominance. Having one copy of an allele is not enough to give you that phenotype. You have to have both. Basically, another way to state a definition for being recessive. The opposite of the dominant allele. Sir?
Are we always going to use plus to represent wild type, or do we change it?
Don't have to. That's convention, that plus is usually, when we use plus minus notation, it's usually because we already know or figured out that plus is wild type and that minus is representing something that's mutant. And if I were ever to change that convention, I would explicitly say so. But typically plus represents wild type and minus represents a non-wild type, a mutant allele. So Punnett Squares, the P word, is all about predicting outcomes of crosses. You've already been doing that today. You've been doing this for years as students, setting up Punnett Squares. That's why so many of you sent me when I asked, draw a graphical representation of the outcome of this cross. Most of you sent me Punnett Squares, even though that's not actually what the question asked for. So that's like, it's habit. We've ingrained, which is great as a genetics instructor. You've been ingrained to draw Punnett Squares. How do you know how many axes to put on a Punnett Square? Why is it a square, in other words? Why isn't it a triangle or a cube? Okay, so you need rows. So why are there only rows and columns? This is going to get a little bit philosophical. Sorry. What is important about the number of axes? That is, why is it a square? What do you put on each axis of a Punnett square? What are the labels of A and B? What do those things represent that you write there? These are alleles. Okay, so what alleles go where I've written A? Which alleles? Whose alleles? How do you decide what to write across the top and what to write down the left side? How many parents are there in this cross? 2. What are their names? A and B. Right? You write the haplotypes of the gametes of one parent on one axis. and you write the haplotypes of parent B down the side, and then you make all the possible combinations. That's what this is a representation of. So does it have to be a square? What would it mean if this was a Punnett cube? What happens if you add a third axis? That means you've added a third parent. Does that happen in nature? Are there any times when 3 organisms have to get together to produce an offspring? None that I know of. There's actually so humans are actually starting to do this in vitro, to create 3-parent babies. Maybe we'll have time to talk about that later in class. So it may be that someday we have to make Punnett cubes. But for now, sexually reproducing organisms, it takes 2 to tango. Hence, we have a 2-dimensional graphical representation of what happens when those 2 individuals mate. There's an x-axis and a y-axis, that's it. So that's one aspect of the Punnett square that maybe has never been explained fully before. I will expect you to use this information later in the term. How do you know how many columns and rows to write? Why is it that when we do a cross, we normally start with a Punnett square that's got 2 columns and 2 rows?
It depends how many genes you're comparing.
It depends in part on how many genes, correct. So in this case, I've drawn a Punnett square to look at the transmission of 1 gene. So let's say it was 2 parents. Parent A is heterozygous and parent B is heterozygous. So we'd make this sort of a Punnett square. What would have to happen to add more columns or more rows? You'd need more, not genes, but yeah, more alleles. So in what sort of organism would you have to add more alleles and more columns and rows to this to represent all the possible gametes that are produced? Something that's not, why are there 2 columns? Why do we have 2 columns? Why do we have 2-car garages? We're diploid. So there's only 2 alleles that can come out of every individual, maximum 2. So for diploid individuals, we use 2x2 Punnett squares. If you were a tetraploid organism, if I asked you on the next exam to set up a Punnett square for a cross between tetraploid organisms, you might have to use something like a 4x4 Punnett square. Because if you're a tetraploid, you could have multiple types of haplotypes in your gametes. I won't do that. Try to put the fear into you. But no. We won't do tetraploid genetics this term. So that's how we know how many rows and how many columns. So diploid, single gene, 2 parents, it's a square, 2 axes. Diploid, 2 columns, 2 rows. You can use this to your advantage. That's why I'm spending so much time on this point in class. Probably too much time on this point in class. You can use this information to your advantage to improve how quickly you solve problems related to setting up crosses. Here's an example. So pretty much every one of you submitted these sorts of Punnett squares when I asked at the end of last class, draw me the Punnett square for this cross. A heterozygote crossed to a homozygote. Okay, some of you put the homozygote on the y-axis. Some of you put them on the x-axis. Doesn't matter. You can free to choose which parent's gametes you put on which axis. Doesn't matter. There's no convention. How could you simplify these Punnett squares with the information that we just discussed? We'll talk about that in just a second. So these are all correct answers. The vast majority of you sent exactly that in. So let's talk about a different monohybrid cross. What happens, before it was crossing homozygotes together. Now we've got a heterozygote. Asterisk over plus. if it selfs, what does the Punnett square look like? If you wanted to draw this as a cross, what genotype would you put next? You're selfing one organism that's a heterozygote. You're crossing it to itself. So what does the Punnett square look like? How many squares in the whole table? 4, so 2 by 2. So this is kind of tricky because it seems like there's only one parent. You can't have a one-axis Punnett square. Well, actually, you could, I guess. So out of this, you get the 1 to 2 to 1 genotype ratio that you probably remember. You fill in across the rows and down the columns with what's listed along the top and the side of the table. Okay. That's genotypes. What are the phenotypes of these worms? What does it take for GFP? We already talked about dominance. What fraction of the total worms that are produced by this one individual when it selfs, what percentage of them will be green? What does it take to be green fluorescent? What genotype? Has to be asterisk over plus. So we can have one copy of the GFP gene. What about the homozygote? Will it be green also? Yeah. So what fraction of the offspring are green? Everybody who's got at least one asterisk is green. So in this case, for example, you could color in, if you want, you don't have to, you could color in the part of the Punnett square where any individual has at least one asterisk, has at least one copy of the GFP gene. That area represents the percentage of the population that has that trait. That area in the table. What fraction of the table did I fill in? 3/4, 75% of the individuals that are produced by this cross will have that trait. That is, when you sum those two numbers together, that's 3 parts out of the 4. So that's how we go, and you may already know this, from the 1 to 2 to 1 genotype ratio to a 3 to 1 phenotype ratio. Because it's both the homozygotes for the dominant gene and all the heterozygotes that have that trait. So genotype ratio, phenotype ratio, 2 different things. It's all from the same set of data. It's all derived from a Punnett square. You just have to know how to convert genotypes to phenotypes. To do that, for the most part, you only need to know dominance. You need to know what's the phenotype of the heterozygote. That's why being able to do those crosses with the pure breeding parents and looking at the phenotype of the F1, you have to do that to figure out dominance so that you know what phenotype those heterozygotes are going to have. That's how you predict what you expect to see in the next generation. It's all about the heads. What happens when you do the cross to wild type? So in whatever fun color this is, where do you cross that heterozygote to? You cross to wild type. It's not asterisk over asterisk, it's the other one.
Plus, plus.
It's plus over plus, wild type, genotype, usually. Okay. What does this Punnett square look like? I'm going to throw you a loop. I'm not going to draw a Punnett square. I know you expected I was going to draw a Punnett square, but I'm not going to. I'm going to draw the Punnett rectangle. Who here has heard of a Punnett rectangle before? Be honest. Anybody? Look around. See? Nobody's ever heard of a Punnett rectangle before. Why is this a Punnett rectangle? Here's one axis. That's the heterozygote parent's gametes. 50% are going to be asterisk, 50% are going to be plus. What am I going to write on the top for the wild-type parent's gametes? Plus. Why the hell would I bother writing the other plus when it's no different from this plus? The width of this column represents 100% of the gametes that plus over plus parent is going to produce. The height of this table represents 100% of the gametes that the heterozygote parent produces. It's divided in half because 50% of its gametes are plus and 50% are asterisk. Why draw extra lines? Make a Punnett rectangle. What percentage of the offspring are going to be heterozygous? 50%. Fill it in. Half of the area of the table. Sure, you could write out the other column. You could put plus in there, but what's going to happen when you figure out what's the fraction of heterozygotes? It's still half of the area of the table. So anytime you have a parent that's homozygous, you don't even have to write that extra column. This will become very useful when we start talking about dihybrid crosses, when we start talking about working with 16 square Punnett squares, 4 across and 4 down. Make it easy on yourselves, skip the work. You don't have to necessarily draw multiple columns when they're the same haplotype. The critical thing to recognize is that the relative widths and heights of these cells is all about the frequency of those gametes. If it's 50-50, the columns will be the same width, the cells will be the same height. There are times when we'll see something else. And then there will be squares that are not squares, they're rectangles. Okay, so we've done a little bit of converting almost last point. One new concept. We've talked about converting genotypes to phenotypes. What information did we need to know the genotype of, or the phenotype rather, of an individual that's a heterozygote? What information would I need to tell you? So we'd have to know something about which allele is dominant. In this case, which allele is dominant by convention? The big A. So what else would I need to tell you? It's the really obvious question that you're probably asking yourself. I heard somebody say it. So big A is dominant. What does it look like? What's the phenotype? Make one up. What's the phenotype? Name a phenotype. Brainstorm with me. Any phenotype.
Brown hair.
Brown hair. Okay. So the big A allele makes individuals that have brown hair. All right. What's the recessive condition? So if big A is dominant, then brown hair could either be big A over little a, or what's the other genotype that would be brown? Big A over big A. Also brown. What's the alternative? Taupe. I don't know. I'm just making stuff up. So let's say the recessive allele gives you taupe-colored hair. That's converting genotype to phenotype. You just need to know something about dominance. What happens if I tell you that you have an individual that has brown hair? Can you convert that phenotype back into genotype? What's the genotype of an individual with brown hair? Can you tell for sure? No, we just said that. There are two different genotypes that produce this phenotype. So as geneticists, we can usually predict phenotype based on genotype as long as we know dominance relationships. But you can't necessarily go back the other way. So if you're given a phenotype, it's harder to go backwards to infer the genotype based on the phenotype. Unless you happen to be one of those rare individuals that has the taupe hair, in which case, what's your genotype? Little a over little a. So you can convert from phenotype to genotype and back. It's often harder to guess somebody's genotype based on their phenotype. So at this point, we talked a little bit about dominance relationships. We used that a lot during class today. And then we've also started using Punnett Squares to look at setting up crosses, predicting the outcomes of crosses. We also used a Punnett rectangle. So here's the last question. So you start with a similar cross to what we've seen before. You've got two dogwood plants. The one on the left is a mutant. It's got leaves growing out of the flower, right in the middle of the flower. The other one's a kind of a weird looking dogwood that's missing one of the leaves. Normally they have 6 leaves. So you make an F1 plant by crossing these two together that looks like this one in the middle. It's got leaves growing out of the flower. So if you take that plant, that F1 plant, you self it, and you see 456 F2s, as it says up there, that also have leaves growing out of their flowers, I'd like you to tell me how many of the plants in that F2 generation look like that. That's the goal. So you should be able to do this with all the knowledge you have now. Punnett squares, dominance. Maybe bring that back for next class. We'll discuss starting then. The other thing is, finally, we get back to a question I posed a week or more ago, trying to combine those roller worms with the green fluorescent worms to make the worms that are glowing and rolly and you sell at raves and make lots of money. Now we can actually start doing this. So next class, this is the end of Mendel's first law, segregation of alleles into gametes, meiosis. Next class, it starts the second law, independent assortment, where we're going to start talking about how to do combinations of different traits that are encoded by different genes into the same organism. So think about this for next class as well, and I'll see you then.